Let's start the lecture. So myself, Kundan Solanki, okay, and I'm a PMRF scholar, and this is a sixth session of cell biology NPTEL course, okay, tutorial session. So in this session, uh, we will be discussing about the chromatin organization, okay, and how chromosomes are organized into from a single DNA molecule into a complete chromosome, how it is packaged. So in that we will be seeing different the protein known as histones. Okay, the histones are the proteins which plays an important role in the folding of the DNA and efficient packaging. So as you already know that the DNA is very long. Okay, if you stretch the DNA into its full length, the DNA is very long in the lengthwise. But due to several proteins associated with the DNA, it helps in the compact packaging of the DNA into a single chromosome. Okay, and that is how it will allow the DNA to package very efficiently in a small size. So we'll be seeing that different types of proteins. So one such protein is histones. Okay, and apart from histones, there are uh, SMC's protein. Okay, SMC protein stands for structural maintenance of chromosome. So that protein includes cohesin, condensins, and all those proteins, which helps in the condensation as well as cohesion during the replication step, as well as condensation of the DNA, which allowing the efficient packaging. Okay, so in this week, if you have gone through the lectures, which was uploaded on the portal for the week 6, so we discussed the discussion was regarding that. So I will summarize first what is the content of that week. Okay, we will briefly go through that week content, and afterwards we will solve some assignment questions, some model assignment question, previous year assignment questions regarding to this content, and also the assignment questions of the previous week. Okay, week 5 assignment questions. We'll be seeing that regarding the meiosis and meiosis process. What were the questions and what could be the probable answer for that? Okay, so I will share my slide. Okay, so is the slide visible to all of you? Can anyone type in the chat box? Otherwise, if it is visible, then we can start. All right. So this is regarding the sixth session. Okay, and today we'll be discussing about as I told, regarding the histone proteins and the packaging of the DNA. Okay, so first things is that you already know that human cell has a genome size of 3,000 mega million base pairs. Okay, 3,000 million base pair is packaged into 23 chromosomes. So 23 chromosomes contain 3,000 million base pairs, and chromosomes are diploid in the somatic cell. Okay, in the zygotic cells, the chromosome is haploid in number, but in the somatic cell it is diploid. So there are 23 pairs of chromosome, and the total length of DNA is 2 meters, and which fits into a nucleus having a diameter of 5 to 10 micrometers. So from the 2 meter, the chromosome has to fix into a 5 to 10 micrometer diameter. Okay, so this size is very small. For that, the DNA needs to be compacted enough so that it fits in that, and that is what that over there the role of histone proteins and other packaging proteins play a role over there. Okay, so to achieve this, the DNA is bound to basic protein termed as histone that helps to package it to fit within the nucleus in a highly condensed form. Okay, and chromatin, the complex of DNA with histone and non-histone protein is termed as chromatin. So the DNA which is bound to histone and other non-histone proteins. So non-histone proteins includes the other proteins which help into packaging, okay, apart from histones. So we will see that there are like condenser and coesins, all the other proteins which are involved. So that are non-histone proteins. Okay? The complete structure that is known as chromatin. And how the histone protein is made, there is four histone proteins which forms an octamer, and around with the DNA is wrapped. Okay, so four histone proteins is wrapped around the DNA, and it makes an octamer structure. So there are four histone proteins make up the eight subunits. Okay, each of each histone protein have a two subunits each, and there is a twice as much protein as the DNA in chromatin. So the number of proteins, that is histone proteins and other non-histone associated proteins, are twice in number compared to the DNA. Okay, so it is how it is there. Okay, the chromatin organization basically at the initial level. Okay, after that there are many packaging involved, and you will get the so you will get the complete chromosome structure. So the most basic level of chromosome organization is the nucleosome. So here you can see the nucleosome. Okay, this is the nucleosome. The nucleosome is basically uh, histone proteins onto which the DNA is wrapped. So this is the DNA which is wrapped onto the histone proteins, and here you can see octamer structure of the histone. Okay, four histone proteins made up in dimeric form. Each histone has two subunits each. And the electron microscopy analysis of the interface nuclei reveals a fiber with a diameter of about 30 nanometer. Okay, so interface nuclei is a nuclei during which the replication starts. Okay, and at the interface stage, generally the chromosome is not compact; it is loose. Okay, because during the replication stage, the DNA has to be unwounded from the histones, and that is how the replication will occur. So interface stage nuclei generally is loosely packaged loosely packaged chromatin structure, and that has a diameter of 30 nanometer approximately. Okay, and when the chromatin is subjected to treatment that allows to unfold partially, it appears as a bead on a string that is 10 nanometer. Okay, so when it is unfolded, so interface nuclei is a 30 nanometer, but when it is unfolded by certain detergents and reagents, so then that structure, there you can see the complete histone DNA wrapped around the histone. That structure you can see. Okay, here we will see further ahead. So how the chromatin complete organization is formed, but that is a 10 nanometer in size. Okay, this structure here you can see. So the string is the DNA, and each bit is the nucleosome core particle. So nucleosome core particle is are histones. Okay, the histones match up the nucleosome core particle in which all the four types of histones are there. The nucleosome core particle consists of DNA wound around a proteinaceous core formed the histone protein. And each nucleosome core particle consists of 8 histone proteins, that is, two molecules of each histone, H2A, H2B, H3, and H4. Okay, so two molecules each of these four histones, H2A, H2B, H3, and H4 are there. In total, 8 histone proteins are present. And the double-stranded DNA that is wounded is 147 nucleotide long. Okay, so the double-stranded DNA that is there, it is 147 nucleotide pair long, wrapped around the histone. So this wrapping around the histone. And the histone octamer forms a core around with the double-stranded DNA is wrapped 1.67 tons. So exactly 1.67 tons. So less than 2 tons. Okay, less than 2 tons are wounded around the histone proteins, and the DNA is packaged around in each nucleosome. Okay, so this is how the nucleosome core particle is formed. So this is the schematic representation of the histone into the nucleosome, and here you can see the H3H4 domain. So H3H4 produce a dimer structure, and then H2A and H2B forms a dimer structure. Okay, so H2 interacts with H2B, And then H3 and H4 interacts with each other and forms a dimeric structure. That dimeric structure, both of the dimeric structure forms a tetramer, okay, H3H4 tetramer. So H3, first there is an individual unit, okay, which interacts with each other, and then another dimer of H3H4 interacts with this dimer of H3H4. So one H3H4 dimer interacts with another H3H4 dimer and forms an octameric structure over here. Okay, here you can see eight proteins of two subunits of each. In the same in case of this H2A and H2B, it also forms a dimer H2A and H2B dimer, and then both of these H2A H2B interacts with each other and forms a tetrameric structure. Okay, H2A H2B tetramer, and then the complete octamer gets formed. Okay, this tetramer H2H2B tetramer plus H3H4 tetramer forms an histone octamer. So that is how the complete histone molecules are formed, and that complete histones are wound to, DNA is wound onto that histone. Okay, and here you can see the tails which is hanging. Okay, so this tail actually of histones plays a very important role in the structural modification of the structural modification of the DNA of the DNA. So based on the histone tails modification on the histone tails, there are the DNA can be loosely packed or tightly packed. Okay, so that depends on the histone modification on the tails. So that is how it is done. And the histones are generally small proteins, which is 102 to 135 amino acid long. Okay, and they have a structural motif known as histone fold. So this fold is called an histone fold, okay, which has a two alpha helix arranged perpendicular and one alpha helix is arranged vertically, horizontally, okay, and that are connected by two loops around each other. So this is how the histone, this is called histone fold, and every histone has a similar structure, and around that only they interact with each other, okay, around the histone fold. And each of the core histone has an N-terminal tail that can undergo several forms of post-translational modification. Okay, the tails are flexible and projects outward. So the H3H4 dimer and the H2H2B dimer are formed from the histone handshake interaction. So this interaction here you see is called the histone handshake interaction. Okay, and that is how it interacts. Each histone interacts with each other. So if the each nucleosome core particle is separated from the next by a linker DNA. So here you can see one histone and another histone. So in between there is a linker DNA present, okay? And that varies in length from a few nucleotide pairs to 80. To 80 nucleotides, so around 80 nucleotides, the linker DNA is there. And the histone wrapped is around 147 nucleotide plus around 80 nucleotides. So total there are 200 nucleotide, okay? Including the nucleosome and the linker. There are around 200 nucleotide present, okay, of each component. So this was reduced by the digestion of chromatin with nuclease that break DNA by cutting between the nucleosome. Okay. So, so linker DNA, because it is a single stranded DNA, it is a DNA molecule around which the protein is not there and no protein is there. So nuclease will cleave that region and there you can get the DNA around which the histones are wrapped. Okay. So this when the linker DNA is cut by the nuclease, here you can get the linker DNA. Linker DNA can be cleaved and how much DNA is attached to the histone proteins, here you can identify. Okay, so after nucleodization, the linker DNA between the core particle gets degraded. Okay, and the nucleus can get cut the exposed linker DNA, but not the DNA wound tightly around the nucleosome core. So the nucleus will cut the linker DNA, okay, but not the DNA wound around the nucleosome core because the protein is attached to that DNA, and because the protein is present, because the protein is present, the nucleus will not cut that DNA part. Okay. And after dissociation of nucleosome into protein, the length of the DNA that is wound around can be reduced, and this protected is 147 nucleotide pairs. So that is how you get to know that how much DNA is attached to this histone protein. So you remove the nuclear linker DNA first uh, with the nucleus, and after that you separate the DNA from the histone proteins by using certain detergents. Okay, and after that the you separate the DNA and and, and you get the complete product, okay, of the DNA in the in the gel electrophoresis when you run. So there you get around 147 nucleotide pairs DNA. So that is how you get the number of DNA which is present. So this is the DNA and the histone proteins bound around it. Okay, and so the histones that are colored in different colors, that is green, yellow, blue. Okay, around that the DNA is wrapped. So this is how the structural organization is there. And here you can see the tails originating from the histone. Okay, these are N-terminal tails. Here you can see so around the each histone. So on this structure, the modifications are done, post-translation modifications such as phosphorylation or other acetylation or deacetylation. All those modifications are done, and that modification actually govern the compactness of DNA. So if certain modification, for example, acetylation, if the acetylation modification is done, then that will loosen the DNA binding with the histone. And if there is phosphorylation, then it will will have the compactness, more compact DNA binding will occur. Okay, so this is how the structure post-translational modification help in the uh, degree of compactness in the DNA. So as I told that various levels of chromatin organization. So this is so that this is a 10 nanometer bead on a string structure, okay, around which the DNA is formed. So this is the most basic structure, okay, of the histone wrapped around the DNA. And then these modifications, these histone proteins are uh, here you can see the linker DNA around with the histone proteins are bound, okay, with the DNA. And then there is further compaction. So 30 nanometer fibers are formed, okay, over here. Each histone then interact with each other and mediate the compactness of the DNA. Okay, and after that, more compactness, there are various other proteins involved, and such as condensin and all the condensin and cohesin. And due to which the more further compactness of the DNA will be generated. Okay, and you get a complete chromosome structure. So this is how the hierarchical organization of the chromatin. Okay. So further packaging into the 30 nanometer chromatin fiber is further packaging into the 30 nanometer chromatin fiber is done by the zigzag model. Okay. So zigzag model here you can see this is called a zigzag model. Okay. Here the arrangement of the nucleosome particle. Each nucleosome particle is around is present in a zigzag way. Okay. Arranged in a zigzag way. That is how the compactness are generated. And the linker DNA that connects the nucleosome varies in length. The different method is further perturbation into zigzag structure. So you may say that why zigzag structure is formed because the linker DNA which is present around the nucleosome, okay, which connects the two nucleosome varies in length. Some might be having 80 base pairs, some might be having 40, some may having 20. So there is not a uniform linker DNA present. So at some place there will be a very compact structure. At some place there will be a very two nucleosome will not be close enough with each other. Okay, so that is how there will be unequal arrangement of the nucleosome during the compactness process. So that is why the zigzag pattern is generally observed during the compaction of the DNA, okay, into a chromosome. And finally, the presence of other DNA binding proteins and the DNA sequences are difficult to fold, interrupt the 30 nanometer with irregular features. So there are, apart from histones, there are other DNA binding proteins as well, which are present on the DNA, which which further inhibits the folding. Okay, which further facilitate the folding, and at some region there will be DNA binding protein, at other region there might not be that protein present. Okay, so that is how, due to that, unequal folding will be present. Okay, and during the whole chromosome organization. So apart from the histone, so there is another histone called histone H1. So in the nucleosome core particle formation, there were four histones, that was H2A, H2B, H3, and H4. Okay, so that makes up the complete histone octamer, and around with the that makes up the histone core particle. But the histone H1 is also present. Okay, and that histone H1 actually binds to the linker DNA. Okay, and it allows the folding. And this histone H1 is larger than the core histone. It is less conserved. So that histone core particle, which contains four histones, are conserved among the species, among different species. But this histone H1 have a different variants of it. Okay, and it is usually less conserved around it. So single histone molecule binds to each nucleosome, contacting both the DNA and the protein. So this histone H1 actually contacts the DNA at the linker. In the linker region it will bind. So around the linker region it will bind, and it also contacts the nucleosome as well as the DNA, linker DNA. Okay, and due to that binding, it further allows the compaction of the DNA. Okay, due to by bridging the gap between the two nucleosome core particles. Okay, by binding to the linker DNA. And it also changes the part of the DNA it exists from the nucleosome. And this change in the exit part is important for compacting the nucleosome DNA to form a 30 nanometer fiber. So if the linker DNA, nucleohistone H1 will bound to the linker DNA, and it will allow the folding. Okay. And due to variation in the binding of the different DNA, it will allow the compaction in the DNA. Okay. Due to bridging the two nucleosome, it will bring the two nucleosome core particles closer to each other, and it will bring the compactness. Okay. Hello. हाँ हाँ हाँ. ક્યારે આવવાનું છે? સર એકચ્યુઅલી મેં સ્ટુડન્ટ છે ને મને આજે બહાર જવાનું, મતલબ આજે મેં નીકળું છું, મતલબ મેં આવ્યો તો ઓફિસે સ્ટુડન્ટ, સરને પણ વાત કરી હતી મેં. તો એને કીધું, એને મંડેનું જ કીધું હતું કે અમારે ત્યાં અહીંયા આવી નથી ડોક્યુમેન્ટ, એમ કીધું હતું એને ત્યારે. તો અમે સાંજે આવી જાય એવું હોય તો, કારણ હા, કારણ કે પછી મેં આજે જ નીકળું છું, તો મંડેના મેં અહીંયા અહીંયા નહીં હોઈ શકે મેં. તો મમ્મી, મારા મધર અહીંયા છે, તો મધર આવી જાય તો ચાલે. એટલે મારું અ, એટલે મેં એસપી સર ને પેલા વાત કરેલી અમે લોકોએ. તો એને કીધું કે તમે ભલે જતા રહી, પણ ઓનલાઈન તમે બતાવી દેજો અને મધરની જોડે સાઈન કરાવી લેજો. હા, તો મેં મધરને મોકલાવી 12 વાગે. ઓકે ઓકે. હા હા, ઓરિજિનલ આધાર કાર્ડ ને એ બધું જોશે ને. હા હા હા, વાંધો નહીં. ઓકે. સોરી ફોર ધ ઇન્ટરપ્શન. ઓકે. Yeah, just a minute. Yes, so this we were talking about the chromatin organization, and we were talking about the histone tails. Okay, the histone H1 histone which binds to the linker DNA, and it allows the nucleosome four particle formation. The binding of the two nucleosome particles with around each other to form a 30 nanometer fiber. And further compaction is done by different protein, that is cohesin and condensing protein, that we'll be seeing further. Okay. So here you can see histone modification. Okay, histone modifications are generally histone modifications. As I told, that is the tail present on the histone N terminal. Okay, so that tail actually plays a very important role in the modification process, and the modification, different modification on the tails governs the compactness of DNA binding with the nucleosome 4 particle. So if certain modifications allow the DNA to loosen, so that it will allow the transcription of certain genes, and certain modification, for example, deacetylation, okay, that will allow the chromosome, it will allow the DNA to compact further. Another is phosphorylation. So how it is done, that I will be discussing. That for example, phosphorylation will help in again binding firmly to that region, okay, binding the making the DNA and histone bind firmly so that the replication will not occur. The transcription and translation of the gene will not occur. Okay. So, for example, in the chromosome as well, there are two types of region. There is euchromatin region and there is heterochromatin region. So heterochromatin generally are the regions which has a genes which does not code for a protein. Okay, so that are transcriptionally repressed region. So they doesn't contain a transcriptionally active gene. But the euchromatin region are generally transcriptionally contains a transcriptionally active genes. Okay, so there also the different types of histone variations will be there. For example, in heterochromatin region, because the genes should not be transcribed, there is no need for a gene transcription. So over there, the histone modification will be less. Okay, or the compactness of the histone binding will be more. There will be a more compact DNA binding with the histone, so that it will repress the transcription. Because in the transcription and replication process, you need the histone to be DNA to be loosened by the histone proteins. Okay, so higher the loosen, higher the decompaction of the DNA, better will be the transcription. So that is in the euchromatin region. Generally, the compactness will be low. Okay, so that is generally governed by this modification. So for example, there are three types of modification here there is mentioned, but apart from these, there are other modification as well. So, for example, acetylation of lysine or methylation of lysine are two modification. On the lysine residue, the acetylation and methylation is done. And also the phosphorylation of the serine residue. Okay, so on the tails, on the serine residue, generally, on the tails, it is phosphorylated, and that will allow the different transcriptional activity of that gene. So some histone modifications can occur just after their synthesis, but before the assembly. Okay, so it is not like that after forming an octamer and binding with the DNA, the histone tail get modified. Some histone modifications are done on the tails previously prior to the nucleosome organization. Okay, and that is generally predisposed. Okay, so this modification will remain throughout. Okay, this modification, that modification, and that will govern the specific function of that DNA around with that histone, that nucleosome particle is bound. So it is not that the nucleosome will bond to DNA and then it will modify. In some cases, the histone tail will get modified before the assembly of the histone of the. Okay. And however, many modification takes place once the nucleosome has been assembled. Okay. So in the majority case, after the nucleosome is assembled, after that only the modification will take place. Okay. And these nucleosome modifications are made and removed by an enzyme known as enzyme in the nucleus. So, for example, histone acetylation. Okay, so for adding the acetyl group, there is an acetyl group, histone acetyl transferase, HADS. Okay, so HADS are the enzyme histone acetyl transferase which add the acetyl group on the lysine residue. Similarly, histone deacetylase, that is called HTAX, will remove the acetyl group. So now, what is the function of acetylation? Okay, so generally the histone is a positive charge. Okay, histone contains a positive charge around its boundary. Okay, around its protein, and the DNA is negative charge. So due to opposite charge attraction, the histone will package the DNA and allow it to bind more firmly with each other. Okay, due to positive and negative charge interaction, it will allow the DNA to bind firmly. But when there is an acetylation of that residue, certain residue that is lysine, so acetylation will remove the negative charge on that histone, okay, on the tail region. So it will neutralize the neutralize the positive charge on the histone basically. Okay, the acetylation will neutralize the positive charge on the histone, and that is how the DNA will loosen. Okay, so if there is an acetylation in the histone tail, then that gene is generally transcriptionally active. But if it is histone deacetylated, okay, if there is a deacetylation of histone, that gene is transcriptionally repressed. Okay, so that is the role of acetylation, that it neutralizes the positive charge on the histone, and it will allow the DNA to loosen from its interaction. Okay, so it loosens the electrostatic interaction between the histone and the DNA. Similarly, phosphorylation generally induce the uh, mediates the more form binding of the DNA. Okay, phosphorylation of the phosphorylation on the DNA, phosphorylation on the histone generally makes the DNA more firmly bound to the histone due to charge, due to changes in the charge between electrostatic interaction between the DNA and the histone. Okay, and similarly on the DNA as well, you can see several epigenetic regulations if you studied. Okay, the DNA as well, several epigenetic regions are there. So that is some regions that is epigenetically repressed, okay, epigenetically silenced by phosphorylation, okay. But in that some region there is an acetylation, and that region is generally acetyl. If there is some region which is acetylated, so that region is generally transcriptionally active. Okay, so on the DNA as well, that modification occurs, epigenetic modification that is phosphorylation and acetylation, as well as in the histone as well. So H-tag generally histone deacetylase. One of the histone deacetylase here is the example given, that is SIR2, which deacetylase H4K16 to initiate gene silencing. Okay. So H4K16 meaning histone 4, and K16 means lysine residue at the 16th position of the histone H4. Okay, so that is how it is denoted, H4K16. So lysine 16 residue lysine of the histone H4 is so these HDAX and all enzymes are very specific. Okay, so it will specifically there are variations of these HDAC proteins, and also for example this SIR2 deacetylase H4K16. Similarly, other HDACs will acetylate different lysine residue on the same histone. Okay, and there are other HDAC enzymes which will acetylate other histone protein and other residue. So single enzyme will not deacetylate multiple different different residues. Okay, so this is the modification. Generally, it is when it is present. Here is the like schematic representation of this modification, and here you can see like methylation, ubiquitinylation, acetylation, phosphorylation, citrullination. Okay, so here you can see the modification. For example, in lysine, generally methylation and acetylation is there. Okay, and generally on the serine phosphorylation, you can see the phosphorylation. So these are the total. Here you can see the residues as well. Okay, total number of residues. Or in H3 histone as we take. Okay, if we take, there are around 36 to 37 N-terminal residues. Okay, 37 N-terminal residues around which different sites it is modified. And in the H4 also there are around 24 residues at the N-terminal tail, and each residue is specific. Okay, at each residue. So this is conserved. Okay, this amino acid which is present around this, it is conserved across the species. Okay, this will not change. Histones are very conserved protein across the species, and the N-terminal tail amino acids also are very conserved. And each and every specific residues, for example, lysine, which is present in the 16th position over here. So histone H4, lysine 16, as we discussed in the previous slide. Okay, around that, at this position, SIR2 will act, and it will deacetylase that histone. Okay, deacetylate this histone tail. These residues are very specific and conserved, and each enzyme has a specificity. around which residue it will interact and mediate the deep post-translational modification. So here you can see H2A and H2B has a two N terminal tail. Okay, so this is N terminal and it also contains the C terminal tail. So in the C terminal tail, you can see the ubiquitination, which is there. Okay, at the C terminal tail, but on the N terminal tail, you not see any ubiquitination. Okay, in all the four histones. So this is one novel thing. One, I mean, one idea which you can take from this image. Okay, that H2A on the ubiquitination only occurs on the H2A and H2B. That is on the C terminal tail. Okay, and on the N terminal tail, there will be no ubiquitination of the histone. Okay, in all the histone core particles. So there you can this unit you can remember. Okay, there is only one site each on the C terminal tail as well on the H2 and H2B. So this is the so why there is a modification? Okay, significance of modification. So modifications of the histone tails have several important consequences. Okay, so why modifications is helpful because it will affect the stability. As I discussed that the modification of the histone tails will affect the stability of the 30 nanometer chromatin fiber and package into a higher order structure. So it helps in the efficient packaging, the modification, and the histone acetylation tends to destabilize the chromatin structure for us because adding an acetyl group removes the positive charge from the lysine, thereby making it more difficult to neutralize the charge in the DNA as chromatin is compacted. So this I told that histone acetylation will destabilize the binding of histone with the DNA because the histone generally has positive charge, and if you lysine generally has positive charge, okay, not the histone. Overall histone is positive, but because of lysine, there will be a positive charge amino group present. So that amino group will be neutralized by this acetyl group. Okay, and because of that, the it will neutralize the binding of the DNA with the histone, and it will loosen the chromatin. Okay, Um, so modify histone tail also can attract specific proteins to a stage of chromatin that has been modified. And depending on the precise tail modification, these proteins can either cause further compaction or can facilitate access to the DNA. So that histone tail which is modified can also attack other proteins. So if certain phosphorylation is there on the serine residues, then only some protein which helps in the compaction or which helps in the other process will it will allow the binding of that. Okay, if there is modification in the histone tail, then only that DNA portion will allow the other proteins to bind. If it is not there, then it will not allow. So this actually act as a signature, okay, on the DNA, on the nucleosome particle. That it signatures, the histone tails are basically act as a signature pattern. So if certain modification there, then it will recruit certain types of proteins. It acts as a signature marker to recruit the protein onto that tail. So depending on the precise tail modification, these proteins can either cause the compaction or the facilitate access to the DNA. Okay, and various combinations of modification are considered. The number of distinct markings for each histone tail is very large. Okay, various combinations are there, and based on that, the different proteins can interact that will allow the DNA to loosen or to compact more firmly. It has been proposed that covalent modification, a given stage can convey particular meaning. So as I told that certain histone modification tails on that, okay, the level of post-translation modification on that histone tail the govern its role as a signature. Okay, it acts as a signature to allow the binding of certain proteins, whether their DNA is transcriptionally active or transcriptionally repressed. So from the histone modification tail, okay, modification of the histone tail, you can get an idea whether the gene is actually active or repressed. So histone code hypothesis. Now, so because this actually tells the histone code hypothesis. Okay, the idea which I discussed that each combination of modification led to a specific protein that will execute the appropriate function. So as I told that with the histone tails, there are certain modifications and that will govern the specific protein, attraction of specific protein on that. So that is called histone code hypothesis. Okay. So from the histone tail, you can say that the protein will interact on that. The protein can interact with this in this region of the DNA. So since the histone tails are extended and accessible even when chromatin is condensed, they allow such modification to be read by reader complex that may then execute the appropriate biological function by attracting additional protein. Okay, so histone tails are extended because the histone tail is generally extended structure. Okay, it is not bound by the DNA. It is a loose structure. It is a hanging structure. Uh, and the chromatin is condensed in that form. Also, the histone tails are accessible simply. Okay, even if the chromatin is condensed, the histone tails are accessible, and due to the modification, it will allow to attract the proteins over there. Okay, to execute its function depending on the modification. There are various histone variants are also present. Histone variants there are present like apart from conserved like H2A, H2B, H3, and H4. There are variants of individual histone as well, which have a different different role during the different developmental stage. Okay, so most eukaryotic organism has a specialized variant histone that are diverged from the main histones on the amino acid sequence. For example, in the sea urchin, there are five histone H2A variants. So there are for H2A variants, there are five histones for H2A. So these variants actually, the variants actually are depends is a variant which has a different amino acid residues on the N terminal tail. Okay, so depending on that variant is considered. So H2A generally the core structure is similar, but the N terminal residues onto which the post transcription modifications are done, that sequence are different. And because the sequence dictates the post-translational modification and recruitment of other proteins, so if you change the sequence, it will also change what type of modification will be done on that sequence, and further it will also change what type of protein will be bound on that histones. Okay, so in the sea urchins during the different developmental stage, there are five types of histones, and each have a different role. Okay, for example, nucleosome that is incorporated this histone may differ in stability from a regular nucleosome, and that may facilitate high rate of transcription and replication that occur during the early stage. So during the developmental stage, at the early stage of the development, there are you need more number of genes to be present in the cells. Okay, more and more transcriptionally active regions in the DNA are there. So over there, the DNA needs to be loosely compacted. Okay, around there, at that stage, there will be different variants of the histone which allows the transcriptionally active DNA. Okay, and at the others during the further stage, uh, 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 at the stage where there is a stability is needed, okay, when there is not much replication need to be done, at that stage there are different variants, and which will allow the compactness in the DNA. And all the histone have a variance except histone H4. So histone H4 is conserved across the species, but apart from that, all the other variants, that is H2A, H2B, H3, okay, all the three histones have a variant present. And also, there is a centromere specific histone, which is a SAMPA. Okay, is a variant of histone H3 actually, and that replaces normal H3 at the centromere in the yeast. Okay, so SAMPA is a variant of histone H3 that replaces normal H3 in the nucleosome of the centromere region. So at the centromere region, generally the proteins, the DNA are very compactly organized. Okay, the DNA, it is a transcriptionally repressed region. It is a heterochromatin region at the centromere, and the DNA is very compactly arranged around that region. So over there, the same pay is there in a variant of histone H3, which allows the other protein to bind and then attract the microtubules. So centromere, as we discussed in the previous classes as well, that centromere is a position around which the DNA arms are, two daughter chromatids are linked attached to each other, okay, at the centromere. And the centromere area only the kinetochore gets attached, and kinetochore will attach the mitotic spindle, okay. Microtubule will attach to this kinetochore, and it will allow the separation. So this region needs to be very rigid, okay. If it is loosely attached, then the strength during with the separation, it will cause a problem in during the mitosis process. This is a very compact area, and the nucleosomes are placed very close to each other, okay. Generally, the same SNP A is present histone, and this actually is recognized by the kinetochore, this histone, and onto the kinetochore, mitotic spindle is further arranged, and that it will lead to the separation. Okay, so this is the this was regarding the histone organization. Okay, how histones are packaged, and further like higher order organization of chromosome as well. By the higher order chromosomes are leveled by the formation of chromatin loops, okay, which are then in the interface cells. So higher order organization of chromosomes are generally present in which the chromatin loops are formed, okay, during the interface cell. And there, in the different cells, like different types of chromosomes are also the different structure of the chromosome. In some case, there is a Lamprey chromosome, which is present in the amphibian oocyte. In other case, there is a polytene chromosome. Okay, so let's see like what is Lamprey chromosome and polytene chromosome. So these are the different organization of the chromosome that we discussed. Okay, so Lamprey is like the homologous replicated chromosome paired to form a highly extended structure containing a total of 4 replicated DNA. So one sister chromatid during the during the S phase, okay, one sister chromatid will be replicated into a two. So generally DNA is a double stranded molecule, okay. So from that double strand, there will be 4 sister chromatids which will be formed, okay. 4 DNA will be formed during after that S phase, and this four structure is actually is not separated, okay. In the Lamprey chromosome formation, these 4 replicated DNA molecules are linked to each other. It is not separated, and it forms a highly extended structure, okay, which is highly active in the gene expression. And and it is very highly active and forms a loop. Okay, it forms a loop around the extended chromatin loops from the chromosome axis that can be seen by the microscope. And a given loop always has the same DNA sequence. Most of the gene presents are being actively expressed. The remaining DNA, which is condensed, is not expressed. So actually, in this case, what is happening is that in the Lamprey chromosome there is certain region which is transcriptionally active and certain region which is transcriptionally repressed. Okay. So first thing is that the chromosome will get replicated during the S phase, and it will form a 4 replicated DNA molecule linked with each other. Okay, it's chromatid. It will not get separated. Okay, with each other, and then there will be a loop formation on the DNA. Okay, so by a single strand there will be a loop formation. In the loop region generally is a transcriptionally active region because the DNA on the loop does not contain any histone proteins. Okay, so because the DNA does not contain histone protein, that DNA is accessible for certain various transcriptional machinery, transcriptional protein. So that region is transcriptionally active. Whereas the region which is not present in the loop, okay, the DNA sequence apart from the loop regions are transcriptionally repressed because that region is actually condensed by the nucleosome. Okay, and that nucleosome actually helps to package the DNA and form the condensed. So that region is generally transcriptionally inactive. So that is how the Lamprey chromosome is formed. Some part are transcriptionally active in the same DNA molecule. Some part are transcriptionally active, and some part are transcriptionally inactive. So generally in normal case, if you want to transcribe the DNA, first you need to remove. First you need to disintegrate the histone, and then the chromosome will get DNA will get transcribed. Okay. But in this case, as the DNA is already formed in the loop way and there is no histone associated, so it will directly transcribe the DNA without dissociating the histone protein. Okay, so that is found in the Lamprey chromosome, and actually this is done because to fasten the transcription process. Okay, because during the histone removal as well, first you need to have the histone modification proteins. After the histone modification, the DNA will get loosened, and then the histone will get disintegrated, and then the transcriptional protein on the DNA will bind, and then it will transcribe. Okay, but if the DNA is already present without histone, then there directly the transcription can occur. Okay, it will save the time, and the replication product will also be more present in that case, because there are very low level of regulation in that case. And another case is polytene chromosome. Okay, so polytene chromosome it is present actually in the secretory cells of fly larva. Okay, that is example in the salivary glands of Drosophila. The multiple cycles of DNA synthesis occur without cell division. All the homologous chromosomes are held side by side, creating a single large polytene chromosome. And the polytene chromosome is a distinct set of dark bands and light bands, which has a genes. So, what happens in the polytene chromosome? It is actually present in the secretory cells of the fly larva, that is in the salivary glands of Drosophila. So, over there, the DNA synthesis will occur, but the DNA will not get divided. Okay, so it will multiple DNA replication will be there. Okay, but the cell division will not occur. So, in a single cell, there will be a many copies of the DNA, same DNA molecule. That is called endo reduplication. Okay, that process is called endo reduplication. Multiple copies of DNA in a single cell. And the DNA that DNA actually are held side by side with each other to form a large polytene chromosome. Okay, the chromosome will form large, so that individual replicated DNA will be form a large structure. And this polytene chromosome actually has a dark band and a light band. So the light interbands are generally the transcriptionally active region, and the dark bands are transcriptionally repressed region. Okay, the band interband pattern reflects different level of gene expression and chromatin structure. Genes in the less compact interbands are expressed more highly than in the more compact band. Okay, so in the interbands, generally the genes are highly expressed, but in the dark band, generally the genes are very compactly arranged, and it is not expressed more. Okay, so during the development, when excitation concentration rises, expression of certain genes coding for the protein that the larva requires for each mold and for the pupation is induced. So distinctive chromosome puffs arise and all puffs recedes. Okay, correlating with the expression of new genes become expressed and old one being turned off. Puffs arise from the decondensation of the single chromosome band and have the chromatin is arranged in a loop-like organization. So this is actually telling how the DNA is getting organized into a single polytene chromosome. Okay, so in certain region there will be puffs. Okay, puffs means the looping kind of region. And in certain region there will be no loop formation. So as I told that wherever there is a loop formation in the DNA, that region is generally transcriptionally active. Okay, and another region which is there is no loop, there is transcriptionally repressed region. Okay, so during the larva stage, when the pupation is induced, when there is a ecdysone concentration rises, so that is a hormone. Ecdysone is a hormone which allows the expression of certain genes, which helps the larva to develop into a pupa. Okay, so at that stage, certain chromosome structure will arrange a pop-like region from where the genes. So, for example, the DNA sequence which codes for a gene which allows the conversion of larva into a pupa, that region will form a pop-like structure so that the transcription will be effective. Okay, the gene will transcribe accurately over that without any regulation. But in the certain region, okay, another region will be transcriptionally repressed. But in the certain regions, for example, there will be a transcriptionally repressed at the later stage, for example, in the development. So the genes which help the conversion of larva into pupa, the puffs present on that will get resided, and certain other genes which were previously transcriptionally repressed now will form a puff-like organization. Okay, which is required at that developmental stage. So POF arises from the decondensation of a single chromosome. So decondensation of that DNA, which was previously condensed, at the developmental stage, it will decondense, and that POF will arise again. Okay, so this POF is dynamic. At one stage, it will at some DNA molecule, at some DNA region, there will be POF formation. At another stage of development, at the other DNA sequences, there will be a POF formation. Okay, so it is dynamic. So this is actually the how the DNA is getting regulated. Okay, the gene expression is getting regulated by the different degree of chromatin organization. So this is the example of that, both the Lamprey chromosome as well as the polytene chromosome. So in mammals generally, in the humans generally, it is not observed this kind of thing. Okay, but this is an extreme example. Okay, I mean unique example which is present uh, for the chromatin organization. It and DNA replication, DNA expression. Okay, uh, but the common expression pattern is that only that histone modification on the tail, and that will allow the transcription of that certain thing. So that is the common type of regulation which is present. And other level of regulation is the epigenetic regulation on the DNA molecule for that is phosphorylation and other modification. Okay, so phosphorylation will silence the DNA molecule. So that epigenetic regulation will be there in the DNA molecule. So then now we talked about the various levels of chromatin organization. Okay, nucleosome 10 nanometer, and that compaction into a 30 nanometer, and then further it is like forming a coil structure. Okay, around the zigzag structure, then zigzag will form a coil structure. Okay, coil structure. So this coil structure actually further helps in the mediation of the compaction and into a single chromosome molecule. How this coil structure is formed? Okay, from the 30 nanometer fiber, how this coil structure is formed? Okay, what are the proteins which are involved over there? We will see. Okay, now we will see that protein. That protein is called cohesin. Okay, cohesin condensing. So let's take a break for 5 minutes, and then we will start how cohesin plays a role in condensation. Cohesin and condensing plays a role in condensation of the DNA molecule. Okay, so let's take 5 minute break. Mhm. Brain cancel series. Brain cancel series. Yeah, so let's start the session. So before starting, like, any one of you have doubt regarding if you have gone through the week? So let's discuss first rather than continuing this. Okay. So any one of you from this week content, if you have doubt, because this session is basically doubt clearing session. Okay. So if you have gone through the week content, so if you have any doubt, you can like type in the chat box as well. Okay. We can answer that. After that, I can start. Okay, for the regarding the regarding what I discussed currently as well. Okay, the histone modification and whether you have doubt in that case, so you can type in the chat box. So we can answer that thing. Okay, yes. Let's discuss about SMC. So SMCs are the structural maintenance of chromosome proteins. Okay, so these SMCs are a class of proteins. The SMCs contains protein known as cohesin, condensin, and other SMC 4-sex complex. Okay, cohesin and condensin belongs to this SMC protein complex, and each and every has a different subunit. Okay, for example, cohesin will have a different protein. Uh, different SMCs are known SMC proteins, while condenser will have a different SMCs are known SMC proteins. Okay. For example, in cohesin, it is conserved in eukaryotes as well as prokaryotes. Okay, and it has a ATP binding domain. So this head region which you see over here, that is an ATP binding domain. It will act as an ATPase. So ATP will bind and it will convert the ATP into ATP. It is an ATPase generally. It will convert the ATP into ADP and pyrophosphate. Okay, and it is a flexible coil region. So this region you can see, and a hinge region over here. Okay, this is a hinge region. This is a flexible coil region, and SMC proteins dimerize with each other. So in cohesin, generally there is SMC1 and SMC3. Okay, so it dimerize with two. Okay, and there are non-SMC proteins as well, such as RAD21, SA, WAPL, and PDS5. So these proteins these are the non-SMC proteins, and these proteins actually mediates the interaction of the two SMC at the two SMC head region. Okay, it dimerizes the two SMC head region by bridging bridging the two SMC dimer, and it is also post-transcriptionally modified by acetylation. That is by ECO1. It is an enzyme which promotes acetylation, and also sumoylation and phosphorylation of that. Okay, post-transcription modification. Okay. And this cohesin, as we discussed, that cohesin, what does it mediate? Cohesin actually binds the two DNA molecule when it is replicated. Okay, so cohesin will bind the two daughter chromatids with each other, so that the cohesin will separate during the anaphase stage. Okay, during the anaphase stage by enzyme known as separase, it will cleave this cohesin subunit, that is RAD21. It will cleave, and then during the anaphase state, that cohesin will get disrupted, and that is how the two sister chromatids are separated. Cohesin are the proteins which help to bind two sister chromatids to each other. Okay. Here you can see the SMC proteins. Here you can see the domain. That is, this is the hinge region, and here you can see ATPase domain. Okay, this is a single SMC protein, and two SMC dimer gets bind with each other. Okay, to form a ATPase head domain. Okay, and this is the hinge region. At the hinge region, another SMC protein will bind, and at the ATPase head domain, the non-SMC proteins will bind. Okay. For example, here you can see the heat subunit. So this is cohesin, in which SMC1 and SMC3 are present. In condensin, generally the SMC4 and SMC2 is there. Okay. It also contains the glycine subunit in the condensin, and another SMC is the SMC5 and 6. So these are the three main SMCs which are present which helps in the chromosome during the chromosome organization. Okay. These three, specifically cohesin and condensin, are the two main SMCs which are important. Okay, during the compaction process. Whereas these SMCs will generally plays a role in the DNA repair process in most of the case. So each and every protein has a different SMC subunit. Okay, in SMC3 and 1 is present in cohesin. There is condensin, there is SMC2 and 4. In SMC56 there is an SMC56 complex. Okay, and other known SMC proteins are also present. So as I told that during the replication, the SMC cohesin generally binds the two daughter chromatids. Okay, and during the anaphase stage, the separase cleaves this RAD21 subunit. Okay, so RAD21 is a known SMC protein which binds these two SMC protein. Okay, and this red separase will cleave this RAD21, and that is how the linkage between the two SMC will get disrupted, and it will allow the resolution of these two daughter chromatids, and it will lead to separation and site separation. That is how it is regulated. So cohesin, as I told, there is having SMC1 and 3, MCD1, SSC1, and SSC3. These are the non-SMC these are the non-SMC subunit, MCD1 and SSC3. And it helps in the sister chromatid cohesion, and it is essential and conserved. So apart from the cohesion, okay, so apart from the cohesion, it also plays a role in the condensation process as well as during the DNA repair process, gene expression, and replication process. Okay, so all these in other ways also this cohesion plays a role apart from the cohesion. Okay, the defects in the cohesion subunit results in the several developmental defects in the human. So if you have the defects in the cohesion, then the two sister chromatid will not remain attached with each other. Okay, and in that case you will have the improper segregation of the chromosome. Okay, the because sister chromatid will not pair bind with each other. Okay, and in that condition you will get abnormalities during the division process. So there is an experiment. Generally, how people know that the cohesin binds to the chromosome. Okay, at which place the cohesin binds. So generally, for that, people do the chromatin immunoprecipitation. Okay, so chromatin immunoprecipitation is a technique for to detect the protein onto which the DNA is bound. Okay, so the region on the DNA onto which the protein is bound. To detect that region, generally chromatin immunoprecipitation is done, and that is also called CHIP. Okay, chromatin immunoprecipitation. So what happens in this case is that you have a DNA binding region, okay, DNA binding protein, and you have a genomic DNA. So that is already bounded with the DNA binding protein. So first you cross-link the DNA, okay, by using formaldehyde. So by using formaldehyde, you cross-link first the DNA complete structure so that the protein and the DNA will remain intact. So during the further steps, this protein will not loosen get loosened from this DNA. Okay, so that for that you need further fixation of the protein onto the DNA. That is for the stage it will not get released. So after that, immunoprecipitation is done. In the immunoprecipitation, basically the specific antibody around that protein. For example, condensin. Okay, so for example, if you take condensin, so condensin specific monoclonal antibody is used that only binds to the condensin and not to the other place. Okay, so in that regard, you can have different antibody as well. For example, here we are talking about condensin. If you want to study about any other protein, so against that protein, you need to raise the antibody. Okay, in the different species, and then you can use that antibody. You can purify that antibody, and that antibody actually will bind to this protein only. So antibody generation also is altogether a different topic. Okay, we will not dwell that into here, but that antibody is highly specific. Okay, that monoclonal antibody is highly specific to a specific protein that will only bind to that protein, not other places. So immunoprecipitation and protein specific antibody is done. Okay, and then you will how do you know that what is the DNA sequence encoding that antibody? For that, you need to reverse crosslink. Okay, you need to reverse the crosslink of the protein with the DNA. You purify that DNA, okay, and you analyze the DNA sequence by the qPCR or microarray hybridization or sequencing method. So from that, you will get to know what are the sequence of the DNA around which that specific protein will bind. So in the case of chromatin, in the case of cohesin, generally you will get two motifs. Okay, so motif one and motif two. The motifs are the region around which the DNA sequence are similar. Okay, so if that motif contains a specific sequence, then only around that region cohesin will bind. So you can get an idea from this that the DNA sequence is this, then around that region the cohesin will bind. Okay, so this study actually is done by the chromatin immunoprecipitation. You can do that study. So this study was done by the Shikha Laloria ma'am only, which is the course instructor of this course. Okay, so this study was done by herself, and they actually deduced the regions around which the cohesin was bound. Okay, the regions on the DNA around which the cohesin are bound, and they found that it has a conserved periodicity. Okay, so cohesin, as you can see, the blue color is a cohesin subunit. It is conserved. Okay, around each periodicity, that is 8 to 9, around every 8 to 9 kb, there is a one cohesin motifs was present on the DNA. Okay, so if this motif is present, then that cohesin will bind at that region only. And the cohesin motifs were highly present in the telomere region. So in the telomere region, they were in the centromere region, the cohesin motifs were very high in number. They detect that the cohesin subunits were very high. Cohesin subunits are very high in number in the centromeric region, whereas at the telomeric region, the cohesin subunits are very low. Okay, the region corresponding to that DNA sequence corresponding to that, okay, that are very low in the telomeric region. And the distribution correlates with the AT-rich region. So if there is a more AT-rich region, there will be higher number of cohesin presence. So this actually also makes sense also because generally the AT-rich regions are very weak. The interactions are very weak, okay, over there compared to the GC. On the GC, you have a 3 hydrogen bond between each nucleotide base pair. So the strength of the GC pair is higher compared to AT-rich region. And the role of cohesin is to mediate the form interaction between the two sister chromatids. Okay, and if the AT-rich regions is high in number, so over there you need more force, more interaction. Okay, so that the two sister chromatids will not get loosened up in that region. Okay, if the more AT-rich region is there. Okay, so that is why the cohesins are present at high number in the AT-rich region because that regions are uh, more prone to separate compared to the GC-rich region. Okay, so that was the observation actually from this study, which was published in Cell Biology into the year 2000. Okay, so you can refer to this paper for more detail. So that is called cohesin associated regions by chromatin immunoprecipitation that was identified. So here you can see the various cohesins and cohesin subunit which are arranged around the DNA molecule. And cohesins, as I told, that it helps in the cohesion, double strand break repair, gene expression, and all those replication as well. Okay. So apart from that cohesin, there is also condensin subunit which contains an SMC 4. In cohesin there was 1,3. In a condensin there is 2,4. Another non-SMC subunits that is cap H, cap D2, cap G. And these are also essential and conserved. And it helps in the condensation process. Okay, the cohesin helps in the cohesion between the two sister chromatids. This helps in the condensation of the single chromosome molecule. Okay, one chromosome molecule. Both the roles are different. Apart from condensation, as we discussed in the cohesion as well, it also plays a minor role in cohesion, rDNA stability. Okay, ribosomal DNA, the DNA present in the ribosome, it also provides a stability for that, and silencing as well, telomere silencing as well as tRNA gene clustering. Okay, in the tRNA generally there is a clustering of genes. Okay, the T in the tRNA. So condension also plays a role in that case as well. There are two condensin complexes in the vertebrates, and the defects in the condensin subunit result in developmental defect that is primary microcephaly. Okay, this is the developmental defect which happens to defects in the condensin subunit. Okay, so this the functions of condensin, as we discussed, telomere silencing, sister chromatic cohesion, condensation, tRNA gene clustering, and stability of the rDNA. Okay. And the condension actually helps to complex the DNA. So it will bring the two DNA strands close to each other. Okay. And the two DNA strands which are apart, it will bring the two DNA close to each other, and that is how the compaction will occur. And in that case, the loop formation will also occur. Okay. The loop formation will occur from that compact DNA. So this is the case. And after apart from that, there is an SMC56 complex. So cohesion and condensate we discussed. There was an SMC56 complex, which also major SNP present. Okay, and they contain SMC56. Okay, and other non-SMC proteins such as NAC3, 4, 5, 6, basically 1 to 6. Okay, and this SMC56 complex helps in the repair process of the DNA. It does not play a role in the cohesion as well as condensation. Only the repair process it will play a role. Okay, and also plays a role in the DNA double-stranded break repair, replication fork stability, and stability of the repetitive DNA. Okay, this is the case. So now this was regarding the general information about the cohesion, condensing, and other SMC complex. Okay, so this was the experimental experiment done by the Professor Ganji. Okay, and which was the content of that also are present on the week 6 lecture. Okay, third lecture, and they did a very interesting experiment actually to see to detect what does condense or what role does condensing plays. Okay, so they did this experiment, and in that experiment, first they purified this condensate protein into individual subunit, and they found this subunit present. Okay, in the condensate, and then what they did was that they immobilize the DNA first. Okay, onto the glass slide, pegylated glass slide. Okay, so pegylated glass slide, it will bind the DNA, and there is an streptavidin biotin interaction. So ends of the DNA are bounded by the biotin. Okay, ends of the DNA are bounded by the streptavidin that will interact with the biotin, and that is how the DNA will get fixed onto the pegylated glass surface. Okay, on the quartz surface. And when you stain the DNA by a dye, so here you can see the visualization of the DNA. Okay, under fluorescence microscopy, here you can see the bright spots observed. This is actually the DNA which are attached onto the DNA onto the pegylated slide. So they wanted to see that what role do condensing plays. Okay, so basically, and what they did, they added a condensing, okay, condensing protein and an ATP. So condenser requires ATP for its function. Okay, without ATP, the condenser will not have play its role. Okay, it will not allow the loop formation because the head region is, as I told, that head region is an ATPase. This head region will utilize this ATP and it will allow the loop formation onto the DNA. Okay, so after adding the condensin and the ATP, here you can see the very bright spot in the middle compared to the other region. Okay, so this very dark spot is actually the loop region forming. So over there, the DNA is getting compact, okay, by the formation of loop. So this actually suggests that the role of condensing is to form a loop and to further compact the DNA. Okay, you can see the local DNA compaction occurring. And when you after the DNA is compacted, okay, and the loop is formed. So if you flow the buffer, so for example, this is an attached DNA, okay, and if you flow the buffers in the horizontal direction, okay, in the vertical direction, you will see how much is the loop is formed. Okay, so this is an extended loop. This was a DNA molecule. Okay, this one, and the loop formation this is extended. So if you flow the buffer on into a vertical direction, you will stretch the loop basically. So this will reveal that how much the loop is formed. That is why flow stretching. Okay, it is called flow stretching. And here you can see the elongation of the DNA. So basically, it is done to visualize the DNA loop. Okay, by condensing subunit, how much of the loop is formed. And here you can see the increase in the size by the active loop extrusion process. Okay, size is increased of the DNA. And this is the real-time imaging by the fluorescence microscopy. Actually, this is a if you go through the lecture slide. Okay, so over there the professor was discussed. I've shown the complete video in their slides. Okay, this is just a image form I have attached over here. So For visualization, you can go through that video, go through that lecture. Okay, there you can will get a better idea over there. So you can see fluorescence in the fluorescence microscopy here. You can see the condensate and ATP. Okay, fluorescence here you can see very bright spot, which is actually tagged. The DNA is tagged with a dye. Then it is observed. The very dark spot is observed. Okay, and after that, this is a further time point. Here you can see more loop formation. So the cohes condensate is making loop formation process. Okay, and then they wanted to check the kinetics of this DNA loop extrusion. Okay, so there was three regions like region 1, 2, and 3. Okay, and they wanted to see whether at what speed the DNA is getting compacted. Okay, so first they took basically have three regions. The green one is the region 1. Okay, in this graph. So the blue one is the region 3, and the red one is the region 2. So region 2 is basically the loop region. So here you can see at the gradual time point increase at the maximum up to 20 seconds. Okay, this is the second time scale. The 20-second here you can see around 30 kilo base pair of the loop formation was there. Okay, from the starting from the 5. So from the 0 to 20-second, there was around 30 to 35 kilo base pair loop formation was done. Okay, by this condensing subunit. And at the same time, the region corresponding to region 1, okay, the DNA corresponding to region 1 gets shortened from 30 to 35 to around 5 kilo base pair, okay, 0 to 5 kilo base pair, which is exactly the opposite of this loop formation. So here you can say that the loop formation on the DNA was taken from this region 1, okay. So region 1 was completely converted into a loop region by this condensing subunit, whereas there was no change in the region 3. Okay, so region 3 DNA sequence was not affected, and the saturation was found at around 20 seconds. That all the DNA was compacted of the region 1. Here also you can see the graphical image. These are multiple. These bands are actually the multiple DNA attached on the slides. Okay, so multiple DNA. Here you can see the same pattern that in the loop size is getting increased in all this DNA, whereas the DNA size which corresponds to region 1, it is getting decreased simultaneously. But in the region 3 DNA, there is no change. Okay, so this was observed in almost all the DNA. So from this, you can derive the kinetics. Okay, that at 20 seconds, there was around 30 to 35 kilo base pair DNA was getting looped by the promases, by the condensate, by the cohesing. So in each second or in each nanosecond, how much base pairs of DNA is getting how much base pair of DNA is getting into full loop formation. There you can calculate from this graph. Okay, so this was the kinetics which was observed, and then they also observed the fluorescence of this condensing subunit. Basically, how many subunits of condensing is getting attached. So this condensing were attached by this dye ATO647N. Okay, and this dye actually binds to the DNA molecule where condensing is bound, and here you can visualize in the fluorescence microscope. There they found that only one condensing was attached to a single DNA molecule. Okay, by counting the number, it was found that only one condensing was associated with one DNA molecule. Okay, there was not many condensing molecule attached. So this was the observation for that, and the conclusion is that DNA loop extrusion by SMC complex they observed first the DNA loop extrusion by the condensing molecule. Okay, and condensing results in an asymmetric fashion during the loop extrusion. So asymmetrically, it will form the loop. Okay, it will gradually increase the loop size, and the single condensing complex is present. Okay, between the two molecules, between in a single DNA. So this was the conclusion. Okay, so is it clear to everyone? This experiment, is it clear? So from this you can get an idea how you can plan the experiment. Okay, if you want to prove a certain hypothesis. Yes. So now there are other portions, small portions left. So what I wanted is to discuss some questions first. Okay. After that we can after that after that we can go through other things. Okay, if the time remains. So this was actually the previous week assignment questions. Okay. So if you have any doubt, then you can ask, and so I will go through the questions and we will see the answers. Answer. We will discuss the probable options. Okay, over there. And if you have any doubt in any of the options, you can answer. You can ask. Okay, in between. First question was this regarding the previous week, that is meiosis. Okay, meiosis process. Pick the incorrect statement about the bivalence. So bivalence, they contain two homologous chromosomes with two sister chromatids. They are linked together by the chiasmata. They appear during zygoting, and none of the above. So what will be the correct answer for this? If you have attempted, then you have already known. Okay. Okay, so all the three options are correct, basically. And regarding the bivalence, so bivalence is the two chromosomes which are paired with each other. Okay, after the synaptonormal complex formation. Okay, when the synaptonormal complex is formed, okay, that is called bivalent when the two homologous chromosomes are paired with each other. So that pairing actually occurs in the zygoting stage. Okay, and it contains two homologous chromosomes with two sister chromatids. They are linked with the chiasmata. They are linked with the chiasmata. Okay, so that is option D is correct. Which of the following diseases is not a consequences of improper chromosome segregation? Alzheimer's, Down, Turner's, and none of the above. So improper chromosome segregation, we had discussed that due to in during the division process, during the mitosis as well as in meiosis, in some case there will be one extra chromosome present. In some case there will be no chromosome present. Okay, so that is the improper segregation, and that will change the ploidy level in a single cell. So what can be the correct option for this? You can turn on your audio and you can answer. Okay, so it will be better for discussion as well if you have attempted this. Correct option is Alzheimer's in this case. Okay, because in Downs and Turner syndromes, as we discussed, it is these syndromes are generally due to the improper chromosome segregation, but Alzheimer's disease is generally mediated through uh, unfolding proteins. Okay, due to unfolding of the prime proteins, that disease is mediated, not by the chromosome segregation. So Alzheimer's disease is the answer in this case. In Saccharomyces cerevisiae, IME2 promotes entry into which stage? So here you have to know what is IME2 first. Okay. So do anyone know what is IME2? Can anyone answer what was IME2? Okay, so IME2 is actually the meiosis specific kinase. Okay. So it is observed in the budding yeast, which regulates the meiosis process in the budding yeast. So in budding yeast, what happens is that during the nutrient deprivation condition, there will be activation of genes, that is IME2, which tells the cells to undergo the meiosis process. Okay, so why in this case if there is really a nutrient depletion condition, then cell generally does not undergo the division process. Okay, mitosis will be inhibited because if the more cell division will be there, then more energy would be spent over there. But the thing in this case is that because in the nutrient deprivation condition, the budding yeast want to have another progeny of the daughter cells which have a better survival instinct. Okay, so in the meiosis generally, due to crossing over, there might be a chance that there will be a there will be a there will be an organism or a daughter cell which have a better survival capacity compared to the parent because of the crossing over occurring due to meiosis. That is the reason the budding is what it does. It undergoes the meiosis process during the DNA sequence exchange of DNA information. There might be a chance that the better survival instinct will be found in the species. Okay, which can handle the current condition daughter cells. Okay, that is why the budding is will undergo meiosis during the nutrient deprivation condition. And over there is a twofold regulation actually happening. So the first regulation is the activation of IME1. So IME1 is a transcription factor which binds to the IME2 gene and it transcribes the IME2. Okay, and IME2 is a meiosis specific kinase, which is a kinase which helps in the promotion of the cells into S phase. Okay, S it will transfer the cells budding is cell into the S phase and it will promote the meiosis process. Okay, so by that it will that IME2 actually will degrade the S phase CDK inhibitor first, the inhibitor which is inhibiting the CDK complex, CDK cyclin complex. The IME2 will degrade that. It will also inhibit the ubiquitin dependent proteolysis mechanism. Okay, and cyclin B, that is cyclin B will get stabilized. The cyclin B, if there is a cyclin B stabilization, then the cell will undergo meiosis. Okay, cell cycle, and the ubiquitin mediated degradation will be happening. That is how it will promote the S phase and chromosome segregation. So this is the roles of the IME2. It will first act on the inhibitor, and it will also stabilize the cyclin B. Okay. IME2 is a meiosis-specific kinase and it promotes the entry into meiotic S phase. So option B is correct in this case. So fourth is classical analysis recognized by which protein? So correct option in this is importin. Okay, so importin is a protein which binds to the nuclear localization sequence present on the protein. Okay, so importin is a nuclear import receptor. Okay, so that nuclear the proteins which contains the nuclear localization sequence will bind to this importin protein, and that importin protein will allow the translocation of into the of that protein from the cytoplasm into the nucleus. Sorry. Okay. So this is how if the protein is not bounded to importin, that nucleus will not allow that protein to translocate inside. Okay. So binding with importin is essential. Pick the incorrect statement about RAN. Okay, so RAN is a protein. So you need to pick the incorrect statement. It is a monomeric GTP binding and hydrolyzing protein. RAN works with importin. RAN can exist in two different conformational states, and RAN is an ATPase. So what can be the correct option? So first you need to understand what is RAN. Okay. So RAN actually is a nuclear import protein. Okay. It is a GTPase. So RAN is a GTPS. Okay, it is not an ATPS. Option D is correct first. So let's understand over here. There is an importin receptor. Okay, importin alpha and beta, two subunits, which will bind to the nuclear localization sequence on the protein and will allow the transfer of that protein inside the nucleus. So in the nucleus, there is a RAN GTP present. Okay, RAN GTP is present, which which acts on this protein and will allow the release of this protein from the important receptor. Because of the GTP activity, it will allow the release. And then this RAN GTP will bind to this important beta subunit, and this important beta will bounded with this GTP will translocate into the cytosol. In the cytosol, there is a RAN GTPS present. Okay, RAN GTPS is present, which will hydrolyze the GTP, RAN GTP into RAN GDP. Okay, and pyrophosphate will get released. And this RAN GDP actually is present in the cytosol. So this segregation is important. That RAN GTP is present in the nucleus, and RAN GDP is present in the cytosol. Okay, if there was a RAN GTP present in the cytosol, then it will it will not allow the important binding with the this protein. Okay, it will directly dislocate the important binding with the this protein. So that is why the segregation is important. So it is a monomeric GTP binding and hydrolyzing protein. So this is correct. RAN works with importin. This is also correct. And RAN can exist in two different conformational states. That is RAN GTP and GTP. Okay. And RAN is an ATPase. So this is incorrect. The nuclear export signals are likely to be reaching which of the following. So nuclear export signals, it contains specific residues. So if that residue is present, then that protein is destined to translocate from nucleus to cytosol. Okay, so leucine, serine, threonine, and histidine. The correct option is option A, leucine. So proteins that are exported from the nucleus have a nuclear export signals which are reached in the hydrophobic amino acid, that is leucine. Okay, and there also RAN, GTP, and GDP proteins are present, but it acts in a reverse fashion. Okay, and the sequence present is rich in leucine. Okay, which translocate the nucleus to the cytosol protein. So pairing of homologous chromosome and crossover occurs during which phase of meiosis? So this is simple basically. It occurs in the prophase 1. Okay, so in the prophase 1, generally the pairing and the crossover happens. And the metaphase, generally the chromosomes are aligned. In the anaphase, the two chromosomes, homologous chromosomes are separated. In the telophase, it is separated into, cytoplasms are divided. Okay. So which of the following is a meiosis specific kinase? This is also discussed. Okay, IME2. IME2 is a meiosis specific kinase which helps in the promotion in the S phase. So other question is, which of the following nuclear body play a role in processing and storage of snRNPs? PML bodies, polycomb bodies, CAHL bodies, and none of the above. So the option is option C, CAHL bodies. So in the nucleoplasm, okay, inside the nucleus there is a nucleolus. Okay, inside the nucleolus there is a nucleoplasm. In the nucleus generally there is a nucleolus. Okay, nucleoplasm. Okay, and there is a chromatin. The chromatin also, there is euchromatin and heterochromatin. So euchromatin is transcriptionally active, heterochromatin is transcriptionally repressed. And there are other proteins machinery as well, that is transcription and replication machinery also present in the nucleus. Okay, which helps in the transcription of the DNA and also the replication. Nucleoplasm, they also contain certain proteins which helps in the for various functions. In the nuclear body, there is nucleolus, polycomb bodies, and CAHL bodies and speckles. Okay. So nucleolus is a site for rRNA synthesis and ribosomal subunit assembly, which is very helpful during the translational process. Okay, ribosomal RNA. Polycomb bodies contains transcriptionally repressed regions. Okay, transcriptionally repressed genes are present generally in the polycomb bodies. And CAHL bodies and speckles generally helps in the processing and storage of snRNPs. So snRNPs actually are produced in the CAHL bodies. Okay, and they are also processed and stored around in that. snRNP's role is generally to inhibit it helps in the regulation of the gene expression, okay, of the RNA, small nuclear RNA, ribonuclear proteins that is present in the Cahill bodies. During which phase of meiosis does clustering of telomeres occur? So clustering of telomeres occur during the zygotine stage, okay, in this case. So now we have like little 10 minutes. So in 10 minutes we will discuss the some questions, some solved questions, okay, like of this class, okay, some model questions from this class. So if you can answer, then you can answer you can answer this, okay. And these are some model questions, not this week assignment question. This week assignment questions are different that you need to attempt, okay. And after that, in the next week we will discuss that. So based on this week, today's content, these are the questions. So each nucleosome core particle is separated from the next by what? Histone tail, linker DNA, linker RNA, and SSP protein. So what can be the correct answer? What can be the correct answer for this? Each nucleosome particle is separated by the linker DNA. Okay. So these are the subunits H2A, H2B, H3, and H4. And these are the nucleosome core particles. And here you can see the DNA bonded. And the linker DNA basically are the DNA which links the two nucleosome core particles. Okay. The cleavage of MCD1 by sorry, sorry. Is there any doubt? Anyone? Yeah. So if you are answering in the comment in the chat box, then I will not be able to see. Okay, because I'm in the presentation mode and I'm not connected with other device, so that I will not be able to see if you are answering in the chat box. So you can turn on your audio and you can answer. Okay, A, B, C, D. You can answer in that way. Cleavage of MCD1 by separase triggers the movement of sister chromatid into opposite half of the cell during anaphase, metaphase, or sister chromatid into a single half during anaphase or metaphase. So what would be the correct option for this? Separase act at which stage generally? So this MCD1 is actually a non-SMC protein, okay, of the in the cohesion which we discussed. So this we discuss at which stage the separase will act. Correct option will be like A, B, C, D. What will be the correct option? You can turn on your audio and then answer. So option A is correct. Okay, this movement of sister chromatid into opposite of during the anaphase stage. It will cleave the SSC1 RAD21 MCD complex. Okay, of the cohesion. Condensation is essential because it prevents the entanglement of chromosome with each other and cleavage of DNA during cytokinesis. It enhances the efficiency of replication. It prevents cell from entering meiosis. So what will be the correct option for this? So it prevents the entanglement of chromosome with each other. Okay, the condensation, the cleavage during the cytokinesis. So during the cytokinesis, you because if the chromosomes are condensed with each other, so that it will allow the efficient packaging into a nucleus. Okay, and during the separation as well, it will be separation will be efficient. If the chromosome is not condensed enough, then there will be a more fiber-like structure, and that fiber-like structure will provide it will make the entanglement of the chromosome, and during the separation stage, it will lead to improper segregation of the chromosome. Okay, that is the role of condensation, that it will prevent the entanglement and cleavage. Okay, it will help in the proper cleavage of the DNA. It will not. It will prevent the cleavage of the DNA during cytokinesis. This is the correct option. Select the correct statement about tabs. Okay, topological associated domains. So this we had not discussed. So we will discuss over here. What is topological associated domains? Topologically associated domains is basically a self-interacting genomic region in the DNA sequence within a TAD physically interact with each other, meaning that DNA sequence within a TAD physically interact with each other more frequently than the sequence within the outside the TAD. So basically, what the TAD is, for example, let's consider that this is a DNA sequence. Okay, this is a whole DNA sequence in which the one enhancer is present over here, one gene is present over here, and another gene is present in that distance. Okay, so in this DNA sequence, which is a single stage. Okay, but in the topological associated domains, due to binding of certain proteins, okay, such as cohesins and other CTCF binding protein, it will bring the two genes closer to each other. Okay, it will form a loop structure and it will bring the two genes closer to each other, as you can see over here. Okay, these two ends are very distinct from each other. Okay, very apart from each other, but due to looping, which is mediated by the protein known as cohesins and CTCF. Okay, these genes have actually this actually brings the gene closer to each other, and that is how. And if these genes are closer to each other, then it will be better. It will have the regulation of the gene will be better. Okay, if these genes are closer to each other. For example, if the enhancer is present over here and over here the promoter is present. Okay, if this promoter is bring closer to this enhancer, then that regulation of this gene, expression of this gene will be better compared to if they are apart from each other. Okay, because enhancer and promoter need to physically interact with each other for gene expression. So that is called topologically associated domains, which helps in the efficient gene expression. So it is not it is not an idea that the topologically associated domains will form in that region where the active gene expression will be needed. In certain cases, if the gene needs to be repressed, there also it will form a certain kind of loops that will make that two genes sequence apart from each other. Okay, so these two genes now are not regulated, will not be able to regulate because the loop region is such that it will not come closer to each other. So these topologically associated domains will also replace the gene expression. Okay, it is not the case that always the gene expression will be active. So this is called topologically associated domains, and the correct TADs are generally topologically associated domains. Okay, and they are present in the interface chromosome. They are generally present in the interface chromosome during the age synthesis phase. Their formation is dependent on SMC complex such as cohesin and condensins, and also CTCF. Okay, so for that protein actually forms the SMC complex. So all the three options are correct. So all of the above. Okay. So is it clear what is TAD? So this is the example. Okay. This was regarding the topological associated domains. Fifth question is histone code hypothesis essentially states that the histone modification is important for the regulation of cellular function. Each combination of histone modification will lead to a specific protein that will then exhibit appropriate function. Specific modification provides stability. So option B is correct over here that each combination of histone modification will attract specific proteins that will then exhibit the appropriate function, as we discussed. Okay, that specific modification on the histone tail will allow that protein to bind, and then further process will get occur. Okay, regarding transcription and all. So each nucleosome core particle consists of two molecules of H2A, H2B, H3, and H4. Okay, along with the 147 nucleotide pairs of double-stranded DNA. So option D is correct in this case. Four histones are present and 147 nucleotide pairs of double-stranded DNA is there. Phosphorylation of histone tails occurred at which amino acid residue? So what can be the correct option for this? We already discussed this. Serine. Okay, so serine is the phosphorylation site. On the lysine, generally the acetylation and methylation occurs. Okay, and serine on the serine generally phosphorylation occurs on the histone tail. This was the site present, and they are conserved. That you need to remember. They are conserved. Each and every amino acids are conserved. What are the main experimental outcomes of double-threaded DNA assay? So double-threaded DNA assay we discussed. that the DNA was linked with the streptococcus biotin complex, and then the kinetics of cohesin was checked. Okay, condensing the role of condensing was analyzed. So to understand the kinetics of DNA binding and modifying protein, this option is correct. To understand the loop extrusion properties of the modifying protein or protein complex, this option was also correct. We also see the loop extension and also the kinetics, how fast the DNA binding was getting done, and to understand directional property of loop extrusion. So by the buffer flow assay, we also checked which direction the loop was getting extruded. Okay, so all these three options are correct. Option D. The last question is, what is the chronological order of micrococcal experiment? Micrococcal experiment actually, what it does is that it brings the two nucleosome with each other, okay, closer to each other. In this, option A is correct. In micrococcal experiment, what it does is that if there is two contacting nucleosome, okay, so these two nucleosomes are not present in a single DNA. Okay, this nucleosome is these two nucleosome which are colored in different colors are of different DNA, but because they are contacting with each other, it will allow the DNA present in this region, in this nucleosome, to regulate the DNA present in this region. Okay, so this is done by the nucleosome mediated regulation. So because of the two nucleosome contacting with each other, it will allow the two DNA to interact with each other and to have gene expression. So sometimes it may be confusing that these two DNA are of different chromosomes. Okay, these two DNAs are not from a single stretch of these two sequence basically are not from a single stretch. Then how this sequence is getting regulated by another DNA sequence which have a different nucleosome regulation. Okay, so to understand that, people did was that they checked what are the contacting nucleosome, and they fixed that nucleosome by cross linker, that is formaldehyde. Okay, and by micrococcal experiment, they degraded the DNA molecules, the linker DNA molecules, and only the nucleosome which is contacted are present. Okay, and the other DNAs are getting is degraded by the micrococcal. And after that, the nucleosomes, the histones are released, and the DNA which are interacting with each other. Okay, they are paired and studied. Okay, so that which genes are regulated by another gene. Okay, so these both the gene sequence are studied, and then the further analysis is done. So in this experiment, basically what they wanted to understand is that regulation of gene expression can also occur by contacting the two nucleosomes. Okay, even if the DNA are far apart from each other, if you bring the two nucleosomes with each other, then also the two genes can get regulated. So there you can see the which DNA are you can get the chromosomal coordinates from this nucleosome content. Okay, here you can get an idea of a map which can have from that you can get the DNA sequence. Okay, which is getting regulated by in this way. This is called micrococcal experiment, which was done. So in that case, first cross-linking is done of the nucleosome, then chromatin digestion is done by the micrococcal nuclease. Okay, and then the ligation is done of the two DNA molecule of the two nucleosome. Okay, then purification of the DNA is done, and the pair end are reads. Okay, those DNA are analyzed, that what is the sequence and the that which chromosomal coordinates it belongs to. Okay, so for example, in the one chromosome, it is fine that in a single chromosome gene can be regulated. Okay, but one chromosome can also regulate the gene in another chromosome. Okay, so that can happen through the nucleosome interaction between the two nucleosome. That can happen. So by the micrococcal experiment, this was proven that two chromosome also can interact with each other, and the gene can be regulated in that way. Okay, so this was all regarding today's class. And if you have any doubts, then you can ask from the today's class. If you have doubt, then you can ask. If you have any questions regarding anything in particular. Okay, so if you don't have any doubts, then we can end the session. And thank you for joining.